Welcome and thank you for standing by. At this time, all participants are in a listen-only mode. During our Q&A session, please record your name and affiliation. This call is being recorded. If you have any objections, you may disconnect at this time. Now I'd like to turn the meeting over to Mr. Mike Strega, Director of the Polar Institute and Director of Global Risk and Resilience Program at the Wilson Center. Sir, you may begin.
Thank you very much, Operator. We appreciate your assistance. And good morning, good afternoon, and good evening, everyone who's listening in from around the globe. This is Mike Strega, as was announced. I am the Director of the Polar Institute and the Global Risk and Resilience Program. And I want to thank you all for joining us this morning for what we call at the Wilson Center Ground Truth Briefings. We use these briefings on issues ranging from day-to-day issues that flare up or of interest around the globe, or to highlight specific programs, specific global issues that we think need quite a bit more concentration and discussion. That fits both of the categories here this morning, a rather interesting program from the Arctic to Africa, emerging research on human-to-wildlife transmission of COVID-19. So I want to thank you again all for participating in this program. This morning's program and the program title reflects a number of realities. First of all, this pandemic is indeed global. From the Arctic to Africa, it goes beyond the human species, and I think it's a stark reality of our globalized, interconnected society, which knows no geographic, political, or social or economic boundaries. As the invitation and highlight on the website indicates, COVID-19 began as a coronavirus transmitted from wildlife to humans, and now research and practical steps on the ground are needed to identify the risk COVID-19 poses to wildlife, mitigate the modes of human-to-wildlife transmission, and protect the species most at risk. From marine mammals in the Arctic, including our beloved narwhal, to the endangered mountain gorillas in Uganda, there are multiple means of transmission that could place these species at risk, with cascading impacts for ecosystems and the communities that rely on them. And I am very pleased to have with us today three leading experts in their respective fields, each adding to our understanding of this virus, its implications, and real-life impacts it's having. So our speakers today, three experts in their fields, Dr. Harris Lewin, the Robert and Rosabelle Osborne Endowed Chair in Evolution and Ecology, Genome Center, Department of Evolution and Ecology, College of Biological Sciences at UC Davis. Dr. Gladys Kalima Tsarskoe, founder and CEO of Conservation Through Public Health, and Dr. Martin Nguyen, Harvard and Case Western Reserve Schools of Dental Medicine and the Marine Mammal Program at Smithsonian Institution. They will speak in that order. I want to remind everyone that's online that if you have a question, not a statement, but a question during this discussion, please hit *1 on your phone, and that will bring you into a question and answer queue. And questions will be relayed to me, and then we will open those either to a specific presenter or to the group. So with that, let's begin, and I will turn the virtual floor open to Dr. Lewin. Harris, the floor is yours.
Thank you, Dr. Sfraga, and to the Wilson Center for this opportunity to discuss our recent research. As Dr. Sfraga mentioned, the preprint of our study has been made available on bioRxiv, and the paper is currently undergoing peer review. Even as a preprint, our paper has generated enormous public interest because of the implications of the results for public health and for conservation of threatened species, which I will discuss during the briefing. I'd like to begin the briefing with some background information that will help you to understand our study. First, as everyone is now aware, the novel coronavirus SARS-CoV-2 is the cause of COVID-19, a major pandemic with more than 3.3 million confirmed cases around the world and more than 250,000 deaths reported in 187 countries. The actual number of infections is unknown, but likely to be many, many-fold greater. So what is known about SARS-CoV-2 infection in other species? So far, it has been shown that cells from just a few species can be infected by SARS-CoV-2, which I'll call CoV-2 for short. These include Rhesus macaque, green monkey, ferrets, and Syrian golden hamsters. In recent weeks, COVID-2 infections have been reported in a Malayan tiger at the Bronx Zoo and in domesticated cats. Rats, mice, and dogs apparently cannot be infected, although there is an unconfirmed report this week of a COVID-2 infection in a boxer dog. And that's about all we know. So where did the virus come from? Research China identified the virus's closest known relative in a horseshoe bat. This virus is called RATG13. However, given the relatively large number of differences between COVID-2 and RATG13, it is highly unlikely that the virus jumped from horseshoe bats directly to humans. What about pangolins? Pangolins carry SARS-like coronaviruses and have been reported as a possible intermediate host. But recent molecular evidence that I will not detail here suggests that this is not the case. To summarize, most scientists believe that the virus passed through an intermediate host and acquired important molecular changes before it entered the human population. However, the host species for COVID-2, before it jumped to humans, has not been established. How does the virus infect humans? It is an established scientific fact that in humans, the cellular receptor for COVID-2 is ACE2, which is an abbreviation for angiotensin-converting enzyme 2. The ACE2 protein normally functions in the cardiovascular system and is important for regulating blood pressure. Now, critical information for our study came from two groups of Chinese researchers who recently reported the three-dimensional structure of the human ACE2 protein complexed with the viral spike protein, those mean-looking, red, spiky-looking things you see in cartoons of COVID-2. A total of 25 amino acid residues that are associated with the viral spike protein on ACE2. The gene encoding ACE2 is found in all vertebrate animals. Therefore, all vertebrates have the potential, in theory, to be infected by COVID-2. I want to emphasize that several other host cellular proteins are necessary for cellular infectivity, in addition to ACE2. The purpose of our study was to use species-specific differences in ACE2 to predict those species that are susceptible to infection by COVID-2 and might have been the intermediate host for the virus before it was transmitted to humans. For our study, we compared the 25 spike binding site sequences of ACE2 in 410 species of vertebrates, including 252 species of mammals, 69 birds, 68 fishes, 17 reptiles, and 4 amphibians, with the goal of identifying species that might serve as a host for COVID-2. By analyzing the similarity of the 25 amino acid binding site residues on the human ACE2 receptor, we developed a method for predicting which species ACE2 proteins would be likely to bind COVID-2 spike, and by inference would be susceptible to infection. We also use other approaches to study the receptor and its evolution to support our findings. I will not discuss those results today due to time limitations. So what did we find? The ACE2 receptors of 103 species scored very high, high or medium, for their ability to bind COVID-2 spike protein. Of these, 41, or 40%, are classified as threatened by the International Union for Conservation of Nature. The ACE2 receptor in all 18 non-human Old World primates and great apes studied, including gorilla, chimpanzee, bonobo, orangutan, and Rhesus macaques, were found to have all 25 virus-binding residues identical, completely identical to human ACE2. Therefore, our study predicts that most, if not all, Old World primate species are highly susceptible to infection by the virus via their ACE2 receptors. And importantly, 12 out of 18 of these very high-scoring Old World primates are on the IUCN Red List. Another group of animals, 27 in all, was predicted to have high probability of binding of the virus by their ACE2 proteins, and thus may also be susceptible to infection by COVID-2. These include 4 species of rodents, but not mice or rats, cattle, sheep, cervid deer, and goats, and several species of whales, including the narwhal, which will be discussed later by the world's leading expert in narwhals, Dr. Martin Nguyen. Of these 27 species, 10 turned out to be on the IUCN Red List, including several species of lemurs and the giant anteater. The Siberian tiger, a close relative of the Malayan tiger at the Bronx Zoo, found to be infected by COVID-2, was found to have a medium score for virus binding to their ACE2. Several other species of felids, including domesticated cats, were also found to have medium risk for binding of the virus to their ACE2 proteins. More research needs to be done to understand whether COVID-2 uses ACE2 to infect cats and what the potential is for transmission of the virus to other animals. Dogs and their relatives, such as wolves and dingoes, were found to have low risk for infection via ACE2. These results do not exclude that the virus can infect dogs through other cellular receptors, but this does not seem likely based on recent studies. What about bats? 37 species of bats were analyzed, the largest sampling ever. Surprisingly, all bat species, including the Chinese horseshoe bat, were found to have very low scores for ACE2 binding to COVID-2 spike protein. These results indicate that the ability of COVID-2 spike to bind human ACE2 was likely acquired in one or more intermediate host species. Our results do not exclude that COVID-like coronaviruses commonly found in bats use bat ACE2. What about other vertebrates? No birds, fish, amphibians, or reptiles were found to have ACE2 that would likely bind to the viral spike protein. However, this does not rule out that these other vertebrates can be infected with COVID-2 using another cellular receptor, as, for example, the MERS does, MERS virus. So what are the takeaways? Our study identified a large number of mammals, 103 in all, that can potentially be infected by SARS-CoV-2 through their ACE2 protein. Our results provide a directed approach for the identification of an intermediate host or hosts for COVID-2, and hence reduce the opportunity for a future outbreak of COVID-19. Of the 103 species that scored very high, high, or medium for the ability of their ACE2 receptors to bind COVID-2 spike, 41, or 40%, are classified as threatened by IUCN. These species represent an opportunity for spillover of SARS-CoV-2 from humans to other susceptible animals, including wildlife, and should thus be a focus of intensive surveillance and conservation efforts. I want to be clear that our results are predictions, and that except for those species that have been shown to be experimentally or naturally infected, all of these predictions must eventually be confirmed experimentally. I'd like to conclude by making 3 additional points. First, I want to emphasize the enormous scientific value of investments made in genomics over the past 3 decades, with rapid, accurate, and inexpensive DNA sequencing methods now contributing to an explosion of data on genes and genomes of hundreds of animal species. Big genome projects such as BAT1K, Zoonomia, the Vertebrate Genomes Project, and the Earth Biogenome Project, which I lead, have made important contributions to the databases of DNA sequences that were used in our study. Second, Our study also highlights the importance of free and open access to data made available through public resources, such as NCBI and GenBank. Thirdly, zoos such as the San Diego Zoo and the National Zoo in Washington, D.C., have been critical in supplying cells and tissues for DNA sequencing and from comparative biology and medicine for studying the host side of things. For many species, especially those that are rare and endangered, zoos are the only reliable source of samples for genetic studies. I'm pleased that two distinguished geneticists and co-authors of our paper, Dr. Oliver Ryder from the San Diego Zoo and Dr. Klaus-Peter Koffli from the National Zoo, are available today to answer specific questions about how information generated by our study is being used by zoos to protect spillover infections from humans to animals. Also, as mentioned before, my colleague, Dr. Martin Uia, will talk about the implications of our findings for cetaceans, especially for those that inhabit the Arctic. Finally, I want to thank Joanna Damas and Marco Corbo, two postdocs in my laboratory, and 16 other collaborators from around the world for working so hard in a very short period of time to produce such important scientific discoveries. And thank you all for attending and for listening to my briefing. And now I'll turn it back to Dr. Sfraga. Can't hear you, Mike. Mike?
Hi, this is Jack Durkee with the Polar Institute. Mike's seen his line drop, actually, so he's dialing right now. Martin, why don't you go ahead and get started with your presentation, and Mike, when he returns, will provide remarks as well. Martin?
Yeah, sure. Thanks, Jack. And thank you, Dr. Mike Sfraga, and the Wilson Center working staff for bringing this discussion to a global platform. My thanks to Harris, too, for laying the groundwork on this pioneering conservation effort that involved a multitude of investigators that worked very hard to produce these results. And greetings to the international community, with a special hello to communities at the Explorers Club, Smithsonian, Canadian Museum of Nature, National Geographic, Harvard and Case Western Universities, and the National Science Foundation and the Prince Albert II Foundation, and our Inuit communities up north, as we learn to listen, care, and remain connected to our planet and the 4.5 billion years of knowledge that she holds. But what exactly are we facing in the Arctic? As background, I've been a field researcher for the past 20 years in the high Arctic, focused on narwhale and particularly the function of its extraordinary tusk. I've been in the water next to them in 36-degree water and assisted on tagging research teams with Fisheries and Oceans Canada, and also have collected Inuit traditional knowledge and anatomical specimens. When we showed in 2014 that the tusk was a sensory organ system, scientists were intrigued and the public was fascinated. Since that time, narwhals have been in the lexicon of more kids than adults. It's the new awesome and cool animal for many. And with that adoration came a new appreciation of the Arctic environment where narwhals live, and the Inuit who observe and know the animal in ways science is only beginning to understand. We know that the Arctic is changing in almost every capacity at 2 to 3 times the rate of the rest of the planet. Changes in the sea, including the loss of polar ice sheet cover, CO2 absorption, ocean acidification, alterations in the chemistry of calcium. Then there are changes on land, including animal crashes like caribou going from 5 million to 2 million in just 2 decades, and other studies on animals continuing under the Smithsonian's Arctic Crashes Project. And then there are changes in the atmosphere, as magnetic north moves at 34 miles per year towards Siberia. So fast, indeed, that the World Magnetic Model, which guides everything from navigation system on ships to smartphone applications using Google Maps, needed to be changed last year to update the reference. So, the variables like animal migration, seasonal change, and ice and weather that give meaning to the 6-season Inuit calendar are all in flux, making an already challenging lifestyle more unpredictable. Of the 252 mammals examined in the paper that Dr. Lewin described, toothed whales were categorized in the high-risk category of binding the SARS-CoV-2 virus. So what does that mean for Arctic species? Well, the Stanford study recently looked at toothed whales like narwhal and beluga and found that they don't actually have the gene necessary to adequately establish viral immunity or defense against many viruses, including this one. And certainly there's an advantage for these whales to have these receptors since, as Dr. Harris-Lewin described, they are involved in blood pressure pathways that can help these deep-diving whales. And based on the high risk for binding the virus and toothed whales not having the needed immunity, certainly I think more study is needed to establish if this risk can indeed happen in the wild and examine the potential modes of transmission that might cause infection. And though we have largely focused on respiratory transmission, we also need to consider wastewater and fecal contamination. The SARS virus can survive 4 days in fecal material and over a week in wastewater. 2 recent medical archive papers cited detectable fragments of the SARS-CoV-2 virus in wastewater systems in 7 European cities over 3 weeks before the outbreak of COVID. And likewise, monitoring of Boston's wastewater titers of the SARS-CoV virus also was revealed recently. And even if there is not a direct link from these wastewater levels to active infection, certainly sewage detection may be a good indicator of what might be coming down. Contamination can occur in multiple ways, with increasing boat traffic, wastewater runoff, and substandard water treatment facilities. And though speculation might assume immediate destruction of such viruses in the Arctic Ocean, we also need to understand that this ocean is quite different than others. First, it's layered with the sub-Atlantic Ocean underneath, and unlike the consistency of salinity in open ocean water, there are multiple bracken water inlets used by migratory marine mammals, as well as glacial runoff and summer ice melt, adding to the fresh water system that can influence marine life and, likewise, viral survival. We all know that our freshwater systems are in peril, as most of the world doesn't have access to safe drinking water. The World Health Organization cites over 2.2 million people every year die of infectious diseases from contaminated water. Our oceans are also suffering. A recent Nature article states that viruses in the sea are a reservoir of the greatest genetic diversity on Earth, with approximately 200,000 different populations of viruses in the world's ocean environment. Now, most of these have restricted hosts and pathways, but the important message is that there is a vast viral ecology, and we are mixing the ocean soup with waste products of a planet now that has 7.8 billion people. So what do we know about the risk to marine mammal species to date? Well, coronaviruses have been found in beluga, bottlenose dolphins, and harbor seals. And yes, viral pandemics and epidemics can be real for marine mammal species. Back in 1988 and 2002, a harbor seal infection of the distemper virus killed over 50,000 animals. So potential risks are real. And as we continue to mix the environmental soup with collected waste and fecal material at one end and consumption needs for everything from food, oil, natural gas, minerals, and building materials, we need to be aware of how we are impacting our environment and indeed this marine environment. So now what do? Do we require scientists working with whales in the wild to undergo additional precautions or testing before being allowed to conduct research? What will be the new normal for zookeepers and aquarium workers? As with all provocative and thoughtful research, insights from the pioneering work of Dr. Harris-Lewin and collaborators give us answers that can lead to better questions. Inuit subsistence hunters are safely consuming their hunt, but do we need to expand the monitoring of potential animal contamination? And what about waste and fecal contamination? How can we monitor new and increasing amounts of waste and associated viruses in the marine environment. We know plastics have made it to the Arctic food web. Should we not be looking at viral contamination sources? These are the next questions we as researchers and conservationists would like to see investigated. Thank you.
Martin, this is Mike Sprague. Yes, thank you for that wonderful discussion. I apologize to all, my line dropped, and so that was one technical issue. The second one is we had trouble getting Gladys connected. She is now connected all the way from Uganda, so I want to thank her for enduring the global technology. I think we have now a wonderful foundation, Gladys, for your discussion. Harris, thank you for setting those, I think, contextual remarks, highlighting the value of the research, what the research is showing, and frankly, a bit frightening as to how this virus not just was jumping from species then into us, but also the implications further down the road. And Martin, thank you for ground truthing this now through our global oceans, but also other species like the narwhal and others. You both have brought up a number of particular issues I'd like to pull a thread on after we open it up for questions, but I think this is a wonderful foundation to now introduce Dr. Gladys Kalima-Sikosuka, the founder and CEO of Conservation Through Public Health. And Gladys, we've had two really nice presentations setting up the scientific findings thus far, and ground truthing now that with Martin in the Arctic area. And now we look at the reach to Africa and what your work is all about. Please explain that to us, what you're finding, what your concerns are, and the work that you will have going forward in the midst of this pandemic. So we welcome you, and I now turn the virtual floor to you.
Okay, thank you very much for inviting me to this panel. Sorry that we weren't able to connect earlier, but at least now I'm here. COVID-19 is something that has really, disrupted the world. I'm going to talk about the work that we do with the mountain gorillas and how we're trying our best to prevent COVID-19 between people and these endangered great apes. I've worked with mountain gorillas for over 25 years, and when I first started working with them, there were about 650. Now they're almost double. There's 1,063. And I was hired as a first veterinarian in the Uganda Wildlife Authority because there were concerns that tourists could make people sick. Gorilla tourism had just begun, and they felt that they needed a vet to stop this. And a disease like COVID-19 is a very good example of the kind of thing they were very concerned about. One of the first things I found when I first started working with the gorillas was they developed a skin disease, and a baby gorilla died, and the rest of the group only recovered with treatment. And this turned out to be scabies, sarcoptic mange, which came from people living around the park who had very little health care. And so we thought, we were only concerned about tourists bringing in a fatal flu, like the COVID, but actually what happened, it was a disease which is common in the community, but because gorillas are not exposed to this parasite, and yet they're closely related to us, it made the baby gorilla die, and the rest only recovered with treatment. And they got it when they went outside the park to raid people's banana plants, because before it was created as a national park, they couldn't really come out, and people used to hunt them. But when it became a national park, people never used to touch the gorillas anymore, and the gorillas lost their fear for people, and now they come out often. So they got it when they went, and someone put out scarecrow with dirty clothing. And we started focusing a lot on improving community health. And we set up, there's been gorilla tourism rules, how you view tourists. But in the past, we have found that people have been breaking these rules because they haven't really seen that they're so important. And some of them from the very beginning were maintaining a distance from the gorillas when you're visiting them. It used to be 5 meters, but it was increased to 7 meters. And you're not supposed to go up when you're sick. You're supposed to turn away to cough or sneeze. That was as far as the rules went. But when COVID-19 pandemic began and we got the first case in Uganda, we got very concerned. We teamed up with the Uganda Wildlife Authority. They asked us to help them to do some training of the rangers. And we decided to upgrade the regulations. So working together with the Uganda Wildlife Authority, Mountain Gorilla Vet Project, International Gorilla Conservation Program, and Max Planck Institute, and the local health centers in the community hospital. We decided to train the park staff. We trained over 100 staff. And the first thing that we told them is that, a lot of the time, having done some research with researchers from Ohio University, Dr. Nancy Stevens and Annalisa Weber, we found out that 98% of the time, people are told that they shouldn't get close to the gorillas. They shouldn't go beyond, closer than 7 meters. But 60% of the time when they were there, if the tourists broke the rules, they got closer than 3 meters. And 40% of the time, the gorillas broke the rules because now they're so used to people and they're very curious. And sometimes they get even closer than 3 meters, so it was very worrying. So some of the regulations we put up were that, anyone with a flu or cough should not be allowed in the forest. And now that sometimes people may not know that they're sick, anyone who has a temperature is not allowed. A temperature above normal is not allowed. The very first index case in Uganda of COVID-19 did not have flu or cough. They actually had a high fever. And then, obviously, hand washing and disinfectant has been stepped up. That was stepped up. We really focused on that. And then wearing of masks became something that was something that people used to think about, but no one really wanted to do it. But now, because everybody's wearing masks and everybody's actually concerned about getting COVID from each other, let alone giving it to the gorillas, wearing masks has now become mandatory. And we were able to find that even if the time that we started doing the training, because Conservation through Public Health, our NGO, joined the National Disease Task Force. And that very day when we joined it, I asked them if they had surgical masks because they're going to do this training. And they said to me that you can't buy them even if you have money because you run out of them, which is quite surprising. But someone from CDC said, actually, cloth masks can work with lining. So we went ahead and contacted a local entrepreneur, right, for a woman, who normally makes tablecloths for tourists, really nice, made up of the local chitenji. And I asked her if she could make masks. And she actually said to me that she was going to lay off most of her staff because she doesn't have any business. There are no tourists. And so she had to tell the women to go home. And I kind of said, are you willing to make masks? She was very willing to make them. And when she made them, she was able to rehire some of the women, which was great. So now all the park staff are wearing masks. This was done with a generous support from International Gorilla Conservation Program. And now we believe when the tourism begins, all the tourists will have to wear masks. So everybody visiting the gorillas has to wear masks. Gorilla tourism was suspended on the 26th of March. And because of disease transmission threats, and of course, having been lockdowns everywhere, it wasn't difficult to actually suspend it. But when tourists return, they're going to have to wear masks, and temperatures will have to be taken before they enter in the forest, and the 7-meter distance has to be really maintained. When we held a practical session the next day, it rained heavily, so I was glad to be wearing actually a cloth mask. And so COVID-19 has enabled us to review the Grade 8 viewing regulations, not only for gorillas, but for chimpanzees as well, and to really upgrade them so that any disease, not only COVID, but any respiratory disease that is harmful to the Grade 8 is not going to result in their extinction or losing them because they're so few in number. Although the numbers are going up, they're still very, very few in number. And another thing that we did is we went back to the community where we've had disease outbreaks in Bimbi and disease spreading from people to gorillas. And we trained this week, we've been training the gorilla guardians, who are people who safely herd gorillas back to the park when they come out. And we've given them all masks, and my team is busy training them to, have the gorillas back safely, be hygienic, how not to give the gorillas COVID, and, turning away to corporate needs, and enforcing the social distancing, which is great. So the human gorilla conflict team are now doing this. And at the same time, we're also going to the village health teams, who are people who we've been training for many years, over 10 years, how to improve on gorilla health, how to improve on community health, how to stop people making gorillas sick, and how to, get people to take up family planning and all these things that improve their quality of life. And so now we're talking to them about how not to give COVID to each other, how to educate their communities to prevent COVID, even looking at for TB patients, because TB patients often have the same symptoms as COVID-19, but also how to prevent spreading to gorillas. So we created some posters also with support from Solidaridad. And so in this way, we are managing to reduce the risk of COVID spreading amongst local communities where the gorillas come out, the villages where they come out, and from people to gorillas at Bindi. So this is what I wanted to say so far about what we're doing. But one other thing I also wanted to bring up is right now there's no tourism, and gorilla tourism has been contributing significantly to the communities around Bindi and to the Ugandan economy. And, for example, 60% of the money for the Uganda Wildlife Authority, which have 22 national parks, comes from gorilla tourism, which is from Bindi Impenetrable National Park and Mugahinga, which is in Virunga. And now with no tourists, it's affecting the Wildlife Authority, it's affecting the local community and all the tour operators. And the local community are not only hired by the park, but they have businesses, they sell crafts, like this lady from Right for No Woman, Evelyn, and her husband. And then they also sell food, accommodation, meals, community walks. It totally transformed the Bindi community when tourism began. And so we are concerned that they have to eat, so what are they going to do next? And there's a big concern about poaching, not only at Bindi, but all the other protected areas where tourists come. So that's the next big concern. But one way that we're addressing it at Conservation for Public Health is we started a social enterprise with Gorilla Conservation Copy 3 years ago. And we found that although most of our customers were tourists who come and stay at the lodges and want to support the community who are growing the coffee, some of them are outside. So we just got an order from the UK. We're also working with John, who set up Pangos.com, and he sells in America, and he's actually run out of coffee. But cargo planes are moving, so that coffee can still go out. So you can actually support the gorillas, prevent the communities poaching or destroying their habitats just by buying coffee. You don't have to actually visit them. So it's an opportunity for people to keep supporting them. And other, things like Right for a Woman, now she's started to make masks. She's also still earning in spite of tourism not being there anymore. And we're trying to look at other ways to support the community. As conservation NGOs, we're all trying to look at all these ways. Since tourism was suspended, how are people going to survive in the absence of tourism? Thank you.
Gladys, thank you very much for that narrative and that description of what's happening there, not just at your particular park and your work, but also how it's impacting the gorilla population, the primate population, but the very communities that rely on programs and conservation efforts like yours. The economic development ripples, the health ripples, the health and safety of the species you're trying to protect, and how that ripples across a broader perspective. Martin, you also teased out not just the very important issue of narwhal and other like mammals, the impact of this virus could be on them, but also the way in which we have infected could infect the environment and how that might work its way into subsistence chains, but also the economic impact and I think the impact of tourism, right? The idea that this virus could stay in either fecal matter or in waste systems for so long, that's a bit of alarming, and then the implications for economic development there, aside from the very important and very real issues related to health. And Harris, thank you again for laying the foundation for all of this, just having us understand better where we think this virus started, how it jumped species, and then got to humans. One question, and I would open it up to the floor here, but one question, maybe either Oliver or Klaus, maybe you could follow up, or Harris, my question is this. So I'll take the speaker's moniker here and just ask the first one. You have all sort of highlighted to me that this virus, like others, can mutate perhaps when it goes from species to species. And I wonder if we're in a vicious cycle here where one species, it could jump from one species to another and mutate, and now we're dealing with that kind of a mutation. And second is now we have a mutation in another species, in another part of the globe. So if it mutates, if this follows, right, if it mutates in Africa or mutates in the Arctic, are now we dealing with a rolling pandemic that's so very hard to catch up with? That is a question for any of you. I'll just leave it there. Who would like to take that rather convoluted question?
This is Harris.
Yes, please.
Yeah, this is Harris, and I'll try to answer your question succinctly in simple terms. There are quite abundant recent data looking at the sequence of SARS-CoV-2, all isolates from all over the world, and the most recent data indicates that the virus actually in human populations is mutating very slowly. And when you look at where the mutations and what kind of mutations are taking place in the virus, it seems that the virus is relatively stable. And what we know so far is that none of, or maybe one of the mutations might affect the virus's function. So I think that's very important. There are strains out there, but none of these strains is apparently more pathogenic than another. Now, the situation in terms of the zoonotic potential, that is something that it could be very difficult to predict. But what I would say is the tools like ours, these genomic tools, and using artificial intelligence and deep learning methods about the nature of the binding of the virus to its receptor, can be used to predict which species will be susceptible if the receptor is known, and if you have a mutation in a known part of the receptor binding domain of the virus, how might that influence binding to a receptor or possibly another protease or another molecule in the host. And so this really demonstrates the incredible value of these genome databases for allowing us to investigate questions related to zoonotic potential of SARS-like viruses.
Thank you for that. Very helpful. I want to remind everyone online that if you hit *1, you will get the first question is from Lena, and I believe is on the board of Wild Foundation. I'd like to ask the question.
Hi, everyone. Thank you so much for allowing me this opportunity. I just wanted to come back to a question concerning the gorillas. When I was listening to the doctor speak, there was mention about people visiting the park, but given how much and the masks that will be used and temperatures that will be taken, but given how much discussion there is about asymptomatic spread, I worry about a great chance of infection if people aren't, in fact, tested. So I wonder if there's a possibility for people to be tested before entering the park. And I ask this question not knowing anything about what test accessibility is like in Rwanda, which I know is an important part of this.
Thank you for the question.
Yes. Well, thank you very much for the question. And it's a really good question. Yes, if somebody is asymptomatic, and actually 80% of people with COVID-19 are asymptomatic. That's what is now being found out. And, if somebody doesn't have a temperature, and even if they're wearing masks, it could be a problem. And, yes, I think the ability to be able to test everybody who comes in could be considered. Currently now in Uganda, a test costs $65. And there's been wide testing, that you do reduce the impact of the virus, and we have very few cases. We only have 83 cases in the whole country right now, and no deaths yet. And they've tested about 30,000 people. And now they're going to test more widely in the community. Still, it's something that we could consider. And that is why it's so important to maintain that 7-meter distance. The one that we found out in the research that people were breaking it. That 7 meters is long enough that even if somebody was, first of all, if you cough into the mask. So that's a much reduced risk to the grade 8, to the gorillas. But maintaining that 7-meter distance means that is a very long distance for you to cough and, spread the virus to the gorillas. So, yes, enforcing the tourism rules, enforcing that distance. Good enough people are used to social distancing now with the whole pandemic. But I think it would be much easier for the tourists to follow these regulations now because everybody has faced COVID-19. They, all over Europe and America, where most of the tourists come from, it had so many cases. So to be so easy to explain to somebody why they have to maintain the distance, why they have to wear a mask, why they're not allowed to go up when they're sick.
Thank you, Gladys, very much. Martin, the circles that we run in here in the Arctic, it's a big area, but it's a big neighborhood, but a small community. So I have friends texting on my personal cell phone a question for you, and that is, if you don't mind, would you address this issue of I'm going to try to boil several down here because I have 4 or 5 coming from friends in Iceland, Greenland, and northern Canada, all about the implications for the subsistence communities in these regions who rely on the ocean for their very subsistence. Can you talk about potential implications here for this virus in the Arctic food chain, specifically, I'm trying to boil down several here, specifically the narwhal?
Yeah, this is obviously going to be a very important issue. It already has become an issue because people remember a lot of the influenza virus carriers, and people's first questions were, is it okay to eat wild birds? Because that was the first order of concern. And I think with regard to the marine environment, we really don't know the transmission mode. We don't know how possible it is, but certainly Dr. Lewin laid out a pretty good format for listing whales as a high-risk community. And we've already known that, in the instance of cats and they having a medium binding capacity, being able to catch COVID, that certainly whales are susceptible and indeed will, be able to likely bind this virus, although that needs to be shown. So we're at a very early stage of understanding the possibilities and potential risks. I would say for now, this virus really hasn't affected a lot of these high northern communities. In Nunavut, where I primarily do the research out of, they just reported yesterday before our call the first case in Pond Inlet, which was released on Canadian News. And nobody knows even how that transmission happened, since it's a very isolated community and the transport is pretty much limited to cargo shipments coming in and out. But I think the short answer is, as I implied with waste, that even if we don't see these as indicators of transmission, these are good ways to monitor viral input into an environment. And we need to start being proactive instead of chasing a problem. So I think for now, people are safe. I don't think this is a concern they should have at the time now for, the hunt that they do and eating subsistence foods. And certainly don't want to be alarmist about that. We want to back away. This is more about, we need to understand more about how we are impacting our environment and the repercussions that may occur, but also not dismissing the science, which Dr. Lewin pointed out, that these whales do indeed have these same receptors and have a high binding capacity to them.
Thank you very much for that. I'm going to condense a couple of questions here and impose on both Oliver and Klaus, because the few that I have here are, what's the role, basically, what has been the role of the National Zoo, the San Diego Zoo, in supporting the very research and the work that Harris pointed out at the very beginning of this discussion. Oliver, can I impose upon you to go first and Klaus second? And again, the question is, what has been your role and perhaps what will be your role going forward?
Thank you. The San Diego Zoo has had a program for over 40 years for banking viable tissue culture cells and biomaterials that we call the Frozen Zoo. And that resource was established for conservation purposes and has contributed to numerous conservation applications for endangered species. But it's also been a resource that has allowed the rapid elaboration of the field of comparative genomics and conservation genomics. So in this era of the greatly accelerated sequencing of genomes, we have been a reliable and resource to contribute samples and help design studies so that we specifically include endangered species, such as in the study that Dr. Lewin mentioned.
Thank you for that. Klaus?
Yes, and for my part, thank you, Mike, is that, it was interesting for me. I was already doing a parallel effort to the study that Dr. Lewin led with some colleagues here at the National Zoo, because one of my bosses at the National Zoo received a request from one of our staff veterinarians to look into this issue with regards to the endangered black-footed ferret, because that veterinarian had seen a study that had shown that you could infect domestic ferrets, and domestic ferrets could develop infections. And so the vet became concerned about what the potential of SARS-CoV-2 might be for a black-footed ferret. So I looked into it, developed sort of a similar type of analysis to what Dr. Lewin's group was doing, but I eventually joined forces with Dr. Lewin because there was just a much more broader base of expertise involved in that collaborative group, which turned out to be excellent. In terms of my part, we contributed a number of, we mined out the genes of the ACE2 receptor from a number, about 12 different species that my group and my colleagues here at the National Zoo, the genomes we had generated from animals, often from the National Zoo. And those were all endangered species. And so with Dr. Ryder and myself, we really thought, this type of research had an important implications and potential impact with regards to endangered species, and especially those species that are managed in zoos like the National Zoo and the San Diego Zoo. And it's had a profound impact here at the National Zoo, because even while at the very beginning of the lockdown related to the pandemic, is that, the zoo staff here, as well as in other zoos around the country and the world, were putting into place, special requirements of, the animal keeping staff and the veterinary staff to interact with these animals, so wearing personal protection equipment. But, our study helped actually to even increase those safeguards to even a higher level, given that, some of the species that were found to be at high risk or medium risk were included a large number, a good percentage of endangered species.
Thank you for that. Appreciate it. We have another question from Lauren Rhesus, who is our Director of our Environmental Change and Security Program at the Wilson Center. Lauren, are you on? Lauren? Maybe she's on mute.
Lauren, if you're on the line, please press *1. Thank you.
Okay, maybe Lauren's not on. Well, we have 5 minutes or so. I want to remind everyone again to *1. I know we've got questions mounted up. Harris, maybe I can just go back to you for a moment. Can you just highlight for us again the next steps? Your report is on our website. It's available through links, and we've provided those as well. can you walk us through the next steps? Because really what I want to do is virtually impose upon all of you. I think that this is something that I'd like to follow up on in 6 months from now, have a follow-up to this call where we can circle back around with all of us online here that have been invited to speak and see where we are. So can you remind us again the next steps in your work and the implications for that?
Yes, sure. Thank you for that question, Dr. Sfraga. The group that collaborated to produce the ACE2 results immediately decided that they would continue, and we would continue, to pursue other genes and proteins and pathways that might be that are amenable to study using the methods that we use for ACE2. And so it is critically important to understand all of the elements of cellular infectivity if one is to understand species differences, not just in whether or not they can be infected, but what happens after they're infected. Will they develop antibodies? Will they develop symptoms like COVID? And will they develop full-blown respiratory disease that is similar to COVID? These are things that are not known in any species except perhaps for Rhesus macaques at the moment. And so it's critically important to do that for a number of reasons. We can understand the basic biology of coronavirus and COVID-like infections, but also it may yield therapeutic targets and vaccine targets. And that is very critical, fundamental research that must be done. Second, on a more practical level, since our study makes predictions about individual species and groups of species, it's very important to test directly and experimentally which cell types, and you don't have to infect the animals themselves. You can do all of this work with, say, cells that are provided by San Diego Zoo. This is why we work closely with Dr. Ryder and others. And obtaining cells that we could look at directly whether the cells can be infected by the virus, and are they very efficient binders, and how much virus they produce, and what types of cells in the animals express the receptors for the virus. So that's a very important issue, especially when it comes to like bats and pangolins, which don't appear to bind the virus through ACE2. The reason that it produces such severe disease in humans is because the receptor is widely expressed in the animal, but most notably on bronchial epithelium and intestinal epithelium, and on kidney cells and heart cells. So many of the symptoms that we see rely on the broad expression of ACE2 in those hosts. We don't know the expression for most of the species on our list, so that also needs to be determined. So these are two sort of big areas that I think must be followed up on, and the people in our group and some of the people on this call will be aggressively trying to obtain funding to pursue these lines of research.
Well, thank you for that. So none of you can say no because we're on a virtual phone call. So let me invite you all back in. We'll arrange something, and I think maybe we'll discuss a particular time, maybe 6 months down the road. You'll all tell me, but I think this is worth it to hear more about the work that Gladys is doing, Martin's doing, and Harris, your team is doing. So I want to thank all three of you. This hour went very, very quickly. I want to thank you all for the work you're doing and the perspectives, not just from the scientific community, but just the communities who are being impacted across the board, again, from Africa to the Arctic, but also the implications for the very species that we often work with and rely upon, frankly. Let me just remind all of those who are still on the line that there is another follow-up to an Arctic-focused program. It's called COVID-19 Impacts in the Arctic, and that is a partnership program on the 19th and 20th of May. It is a Zoom virtual symposium, 2 days, 5 hours per day, and that's a partnership between the Wilson Center, the U.S. Navy War College, and the United States Arctic Research Commission. Again, 2 days, the 19th and the 20th, from noon to 5 Eastern via Zoom. We have information up on the Polar Institute's website, as well as the Navy War College and the U.S. Arctic Research Commission. That will be a tour de force of speakers focused on the impacts of COVID-19 in the Arctic. I want to thank all of our speakers today for bringing us this fantastic discussion. Thank all of the people online who listened and participated. And again, we'll look to a follow-up coming up maybe 6 months from now or so. So I thank you all for listening, and my thanks to a wonderful group of experts. We appreciate very much you sharing that with us. And with that, I will say have a good morning, afternoon, or evening. Thank you, everyone.
Thank you, Dr. 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 Thank you, Dr.